Today's topic is cell lines. What is meant by cell lines? Cell lines are the propagation of culture after the first subculture. What is meant by cell lines? Cell lines are the propagation of cultures after the first subculture. Means after the first subculture, whatever the culture made, that process we'll call it as, or the process or the cells which were cultured are called it as cell lines. First subculture. In other words, we can also say after primary culture, whatever the culture made, subculturings, this is said to be as cell lines. Cell lines have different generations, like if this petri dish is filled, these cells are transferred to second petri dish, and if these cells were filled, these cells were transferred to third plate. So in each plate, the cells will be showing different characteristics. The cells with the different characteristics is said to be as a cell lineage. The cell lineage can be selected by cloning or physical cell separation. So the cell lineage which was separated from these generations is said to be as cell strains. The cell strains do not have infinite life, can die after few divisions. This can die after few divisions. It do not have any infinite life. Can die after few divisions. Okay. So now we will see the types of cell lines. Cell lines are having two types. One is finite cell line and the other one is infinite cell line. Finite cell line will be having a limited lifespan. Means for after few generations, after few divisions, or after few culturings, the cells may die. And infinite is a continuous culture or continuous cell lines, and this is immortal. You can simply say finite is mortal and infinite is a immortal. We will see in detail about finite cell lines. Finite cell lines are the cell lines with a limited culture lifespan. These have limited culture lifespan. It divides only a limited number of times before growth rate declines and then eventually it dies. So limited culture lifespan it has, and it divides only a few times before it dies. Normal cells generally divide for about 20 to 100 times. It depends on species. How many times it depends on species and cell lineage differences. Cell lineage differences and culture conditions. So it depends on species, cell lineage differences and culture conditions. The cells, how many times it can divide, it depends on all these things. For example, if we see human cells can divide up to 50 to 100 times, whereas urine cells can divide 30 to 50 times before it dies. Okay, so finite cell lines are mortal. They die after some divisions. Next we'll see about Infinite cell lines. Infinite cell lines are also known as continuous cell lines. These are also called as continuous cell lines. We can also say these continuous cell lines are transformed, immortal because it has no death, and tumorogenic because it divides rapidly. Some cells in primary culture are altered and becomes immortal. So the alteration we call it as transformation. When it is altered, it becomes immortal, means it do not have any death, it becomes cancerous. It is tumorogenic. These cells have unlimited life, and treating primary culture with some chemical carcinogens like it becomes tumors. Why it is having unlimited life? Because there is some treatment undergone to the primary culture which makes them immortal. And what is the treatment? Treatment may be with the chemical carcinogens. Chemical carcinogens. Carcinogens are cancer causing agents. Next, injecting oncovirus also leads the cells to become immortal. So these two are added to the primary culture to make them continuous cell lines. Next we will see the nomenclature of cell lines. Nomenclature is giving names for their identification. Cell lines can be identified by their names. For example, nomenclature is naming. Naming of cell lines is the nomenclature of cell lines. So, for example, we take NHB 21. NHB indicates the cell line. The number 2 indicates cell strain. Number 1 indicates clone number. So all are named in this order. The cell line here is NHB indicates normal human brain. So from normal human brain, these cell lines, the cells are isolated and these cell lines are prepared. So the strain here is number 2 and the clone number is number 1. And for this nomenclature and for the records in the laboratory, log book is maintained. For the cell lines, log book is maintained to avoid confusion. And at the time of publication, after the discovery, at the time of publication, code is given. During publication, code is also given for these cell lines for their identification. And the code is based on the lab it was prepared. For example for the labs, NCI National Cancer Institute and WI Wistar Institute. So these are the labs where the cell lines were prepared. Next we will see the selection of cell line, how the cell lines are selected for particular experiments. Cell lines, to select cell lines, there are some factors needs to be considered. So what are those factors? We'll see now. First factor is species. So So species non-human cell lines have a less risk. Mostly non-human cell lines were preferred. Next, finite and continuous cell line. Whether we have to take finite cell lines or continuous cell lines also we have to decide before selecting them. Okay. Next, finite cell lines also have some advantages, and continuous cell lines also have advantages. Continuous cell lines can grow faster, and it is easy to clone and easy to maintain, and it gives high yield. Okay. And if we see the finite, it gives the appropriate or right, it expresses right and appropriate functioning of the cells. So it gives the exact cell lines. Okay. So some people also prefer finite cell lines. Next, we have to consider whether the cell lines are normal or transformed. Whether the cell lines are normal or transformed. Whether if we want to prefer immortal cells, then we prefer transformed. Next, fourth is availability. Which cell lines is available in that area, we consider the availability of the cell lines also. Next, growth characteristics. Growth characteristics also considered while selecting cell lines for the experiments. Here, growth characteristics like population, doubling time, means how rapidly the population doubles, and ability to grow in the suspension, how the cell lines can grow. Growth also is considered. Next, saturation density means how much yield it can produce per flask. Saturation density is also considered, and cloning efficiency. Cloning efficiency. So these all things are considered under growth characteristics. Next, stability is also seen, whether the cell lines are stable in the culture or not. Next, how it is expressed phenotypically. Its phenotypic expression is also considered before selecting the cell line. So these are the factors which we need to consider before selecting the cell line. What are the factors we see here? Species, finite and continuous, or finite or continuous, whether we have to select which one we have to use for the experiment. Next, normal or transformed. Next, availability of the cell lines and growth characteristics of the cell lines, stability of the cell lines in the media, and phenotypic expression also we consider before selecting the cell lines. Next, in the next class we will see about a stem cell culture.